Hey guys, we are starting chapter 5. We will be doing section 1 in this video, which covers the structure and function of the plasma membrane. Now, I forgot my headphones at school and I'm recording this at home, so you may hear Jack barking or some loud yee-yee trucks driving by. So if you do, just ignore it. So let's just dive right in. The plasma membrane structure and function. The plasma membrane is common to all cells. Every single thing that is alive has a plasma membrane. It separates the internal cytoplasm, so the stuff inside the cell, from the external environment. Allows sometimes incompatible chemical reactions to occur simultaneously. So it sections off parts of the cell and allows reactions to occur in one part but not the other. Now, the fluid mosaic model accounts for the structure. That is what scientists refer to the plasma membrane. It is made up of a bunch of different parts, which we're going to discuss in a little bit, that are able to float and move as needed by the cell. So it is made up of a phospholipid bilayer. You should remember the structure of phospholipids from the last chapter. Okay, they've got that polar head and those hydrophobic tails. The head, that polar part, loves water. The tails hate water. So the cytoplasmic surface is lined with hydrophilic polar heads, as is the external surface of the cell. Now, between those two heads, there are fatty acid tails, and they're kind of sandwiched in between. Components of the plasma membrane. There are three. There is a lipid component referred to as the phospholipid bilayer that we just talked about. There are protein molecules that kind of float around like icebergs on a sea. There are two types of proteins. They can be peripheral, so that they are only seen on one side of the membrane, or integral, where they go all the way through both sides of the membrane. And the third part is cholesterol, which affects the fluidity, or how easily it flows, how stiff or loose it is. So here is a diagram. Here you can see a phospholipid. We have an integral protein going all the way through both layers, both phospholipid layers, and peripheral proteins down here. They are on only one side of the membrane. So, you may have noticed some carbohydrate chains right here. Okay, they're there for a couple different reasons. There are two kinds. They contribute to the cell's fingerprint. They help identify it. They make it so that cells can distinguish between one type of cell from another. So glycoproteins are proteins that are attached to a carbohydrate chain. So glyco, like glucose, protein, protein. And then the other G is glycolipids, which are lipids attached to a carbohydrate chain. Once again, glyco, like glucose, attached to a lipid. These carbohydrate chains exist only on the outside of the membrane. This makes the membrane asymmetrical, and it makes it so that you can tell which side of the membrane you are looking at. So here you will see the glycoproteins, chains of sugars attached to a protein. Okay, we have a protein. This is an integral protein. We have peripheral proteins here and here and here. Here we have a glycolipid. Okay, we have a chain of sugars attached to a lipid. You'll notice that these chains are only found on one side, the outside of the membrane. You will also notice that peripheral proteins can sometimes team up with integral proteins. All right, and I'm sorry guys, I was wrong on the previous slide. That structure that I pointed to before was not a sugar, that was actually a cholesterol. My bad. Ignore that, delete that from your memory. My bad, guys. Sorry about that. So functions of the plasma membrane. Proteins, channel proteins, they allow the passage of molecules through the membrane via channel proteins. They act like a gate. Carrier proteins, they combine with the substance that is being transported and they assist it through. It's a little bit more choosy. It is very specific about the type of molecules that it will allow through. And then there are cell recognition proteins, like glycoproteins, that help the body recognize foreign substances. Receptor proteins, they bind with a specific molecule, and that allows the cell to respond to signals from other cells. This is how they talk to each other. Enzymatic proteins, they carry out metabolic reactions directly, so they do some kind of chemical reaction. And junction proteins, they attach cells to each other. They make it so that they hold close to each other so that they can form tissues that can then join into organs and then organ systems and so on. So here we have a channel protein. You'll notice that it's just an opening that allows a specific molecule to flow through unaided. It's like a gate. It's like an open door that things can go through willy-nilly as they please, like when I prop the back door open so that Jack can come and go whenever the weather's nice. Cystic fibrosis is an inherited disorder that is caused by a faulty chloride channel, so a channel that looks like this that allows chlorides through, and it doesn't function properly. And this causes a thick mucus to collect in airways and in the pancreatic and liver ducts. And basically they get full of mucus and their body can't process it, and eventually they die. Here is a carrier protein. It is selective and interacts with a specific molecule or ion so that it can move through the plasma membrane. A faulty one of these has been linked to the cause of obesity in some people because they're not able to properly move those ions and it's hard for them to gain energy. This is a cell recognition protein. You'll notice that it has some sugars attached to it, so it is a glycoprotein. And they're different for each person. That is how a person's body, how its immune system recognizes foreign objects, which is why organ transplants are so dangerous. A good friend of mine, his name is Ray, had a liver transplant, and he is on medication for the rest of his life to suppress his immune system, because if it recognizes this sugar chain doesn't belong to the rest of his body, it will attack that liver and kill him. It will stop the liver from functioning, the immune system will attack, and he will die. Receptor proteins, they are shaped in a way that it is so that only one specific molecule can bind to it. Pygmies are short not because they don't have enough growth hormone, but because their plasma membrane doesn't recognize that growth hormone, and so the cells can't interact with it. Enzymatic proteins, they catalyze a specific reaction, and hopefully you remember the term catalyst from chemistry class as something that lowers the activation energy or allows a chemical reaction to occur. So this one is a membrane protein, adenylate silase, I don't know if I'm saying that right, and it is involved in the metabolism of ATP. Cholera bacteria release a toxin that interferes with this protein's functioning and causes sodium and water to leave the intestinal cells and cause very, very severe diarrhea and dehydration. That sounds like absolutely no fun. Junction proteins, also known as tight junctions, they join so that tissues can fulfill a function, so that they can work together, like when a neural tube during development is forming. So without these junctions, without these joinings of cells, Cooperation between cells would not be possible, so an animal embryo would have no nervous system, because the whole job of a nervous system is to communicate with each other, for the cells to communicate. So these tight proteins, these junction proteins, are very important. So here is a summary of all of the different types of proteins. This can be found in your book, and each individual one should be found on a page in your notes. How cells talk to one another. Signaling molecules serve as chemical messengers, allowing cells to communicate with each other. Cell receptors bind to specific signaling molecules, and once that signaling molecule has been absorbed, then the cell receptor binds a cascade of events to occur that elicits a cellular response. It's like dominoes. So the receptor binds with the protein, and it causes one thing to happen, that causes another to happen, that causes another to happen, and it just keeps on going. Signal transduction pathway. An example of this, the pancreas releases the hormone insulin, which is transported to blood vessels in the liver. This causes the liver to store glucose, blood sugar, to take the sugar in your bloodstream and convert it to glycogen. If your body can't do this properly, it's a medical condition called diabetes. You guys have probably heard that. People with diabetes got to watch their sugar because their body can't process it properly. Here is another diagram showing cell signaling. The molecule binds to the protein that causes a receptor to be activated, that causes something to happen, that causes another, and it causes a cellular response. It alters the shape or the movement of the cell, alters the metabolic function, alter gene expression, depending on what the signal is. So take a moment, study this diagram, and I'm going to move on. So the permeability of the plasma membrane. It is selectively permeable, which means it allows some substances to move across it and doesn't allow others. Small, non-charged particles or molecules like carbon dioxide, oxygen, glycerol, alcohol, these non-charged molecules can move freely through the membrane by passing right through those phospholipids. And these molecules follow their concentration gradient. They move from an area that is highly concentrated to an area that is very lowly concentrated. And it'll keep moving until it reaches equilibrium, which means that there's the same concentration on both sides. The movement will continue, but it will be even. Plasma membrane structure and function. Slide 5. Permeability of the plasma membrane. Water moves through the membrane, but it needs some help. It needs help from a special protein called an aquaporin, which its only job is to transport water. The movement of ions and polar molecules across the membrane needs the help of a carrier protein. If not, they'll get stuck in the phospholipids, and they can move through there, but it happens very slowly. Some molecules will move against their concentration gradient, meaning they'll move from a low concentration to high concentration, and that is something called active transport, which means it requires energy. Bulk transport is the way that large particles enter the cell or exit the cell. Exocytosis is when it leaves, sounds like exit. Endocytosis is whenever a large molecule moves into the plasma membrane, into the cell. And we're going to talk about that in more depth very soon. Here is a diagram. We have some charged ions and molecules. All right, they can't move through. We've got some water that can move through. It moves through that aquaporin. Non-charged molecules, they can just go straight right on through. And then large macromolecules, they can't move through easily on their own. They have to have some kind of help in the form of a vesicle that will then fuse with the membrane and allows them to pass in. Here we have a table. You may want to pause the video so that you can get it all written down. Diffusion. The molecules move toward a lower concentration. They move with their gradient. Facilitated transport toward a lower concentration, and it needs help from proteins. Active transport, it moves to a higher concentration, and it requires energy. And bulk transport uses a vesicle. And that is it, guys, and we are out of time, so if you have any questions, feel free to shoot me an email. I'll see you guys in class tomorrow.